Uh, Ladies and gentlemen, now we'll move on to the next talk by Dr. Manjusha. She's already here. Dr. Rohit Sharma, please.
Dr. Manjusha is a microbiologist with a PhD from Cochin University, and she has over 15 years of teaching experience. She began as a guest faculty at Union Christian College and has since worked in various academic roles. Currently, she is an assistant professor at Kerala University of Ocean Studies and Technology and serves as a course coordinator of marine microbiology. Dr. Manjusha has completed major research projects and has extensive publications in her field. Her research focuses on marine microbial diversity, particularly mangrove yeast, and she has contributed significantly to the study of biotic potentials. We welcome you, Dr. Manjusha. The floor is all yours. Please go ahead with your talk.
Thank you, sir. I share my screen.
Um,
Am I visible? Are the slides visible, sir?
yes, Dr. Manjusha. The slides are visible. If you can make it to full screen, yeah.
Good evening, all. Actually, it is really humbling to be amongst stalwarts in the field of marine fungi and presenting a paper like diversity and bioprospecting potentials of mangrove yeast. They have been working decades on their field, but my area, when compared to them, it's just been a few years in this field. And like Dr. Professor Sharma pointed out in the morning, studies on mangrove yeast are much rarer. So here I'm to present my paper, Diversity and Bioprospecting Potentials of Mangrove yeast, and I'm from Kerala University of Fisheries and Ocean Studies, Kerala. Now, this is the overview of my presentation. I'll be giving an introduction to mangroves. I know most of you who have been there in the morning would have heard of mangroves, but this is for the benefit of those who join in the afternoon and youngsters in the group, and a little bit on yeast and how do you go about identifying them, the diversity and distribution of mangrove yeast and their bioprospecting potentials. Now, mangroves, otherwise known as mangle, are intertidal wetland ecosystems found in the tropics and subtropics. They are one of the most prolific and dynamic ecotones. They are abundant in food, a calm shelter to diverse groups of organisms like fishes, invertebrates, and so on. They are a carbon sink, a great role in controlling climate change. They are known to protect the coastal uh, ecosystems. They protect the coast against natural disasters like cyclones and tsunamis. They are biodiversity hotspots, site of tourism. Uh, they are actually locations where a large number of migratory birds take shelter, and so on and so forth. And what is unique about these ecosystems are that the vegetation that are found are known by the same name, that is mangroves. And some of these adaptations you might have heard in the previous talks by the experts. So I would like to just add to some of them, which we might not have heard in the morning. Mangroves, these mangrove vegetation, as you know, are unique. So they are something between the terrestrial and marine ecosystems. And because of the harsh environmental conditions that prevail in these mangroves, they need to be adapted to survive in these conditions. Some of these adaptations are the prop roots or the stilt roots that keep the leaves or the major part of the plant above the water surfaces or protect them from inundations. You have these impermeable roots that prevent the salt entry. You have the pneumatophores or the breathing roots that help them to survive under the anoxygenic conditions in the sediments. You have the leaves that have salt that help in glands that secrete out the excess salt, and you have drop roots that help to collect the excess water, that rainwater that drips off the main branches and so forth. And their flowers are also very attractive, helping in pollination. Their seeds are adapted to float, and actually they are viviparous. That is, they germinate even before they fall on to the sediment. So these are some of the adaptations that these unique, and the leaves are actually very waxy and thick and prevent the loss of water from leaves, because these plants know very well how important fresh water is for their survival, and the very filtration systems that these roots have been the basis of various studies. So moving on to the mangrove map, the distribution of mangroves in the world, you can see the tropic and subtropical regions in different continents. Of course, except the poles, you will find mangroves in almost all continents. And especially you find them in the deltas of the larger rivers of Asia. Now, when you take the picture of India, almost all the South Indian, the coastal states of India have their share of mangroves. And India has around 6.8% of the total mangrove coverage, with the Sundarbans and the Pichavaram being one of the richest mangroves. So my area, as I already mentioned, I'm working on yeasts from mangroves. So when I use the term yeast, the picture that comes to the minds of the listeners are properly this. I hope you would agree. This is the image that comes to your mind, and some of the names of yeast that again strike you are like Saccharomyces, Candida, Cryptococcus. Some of the names probably you have already heard. But when I say Manglicolis yeast, at least from some, this is probably the expression I might expect. So already I have mentioned mangroves are a treasure trove or a hotspot of both macro and micro and macro flora. So are they a rich source of microbiota as well. And among the different groups of microbes that you see, yeasts are one among them. And yeasts that reside in these mangroves are referred to as Manglicolis yeasts. Yeasts basically, they fall under two phyla, the Ascomycetes and Basidiomycetes. And basically, they are saprophytic. There are forms that are mutualistic, commensals, or even parasitic. So when we look back at the history of marine yeast, the first marine yeast was recorded from Atlantic Ocean way back in 1894 by Fisher. But it not almost took 100 years for mangrove yeast to be isolated, and this mangrove yeast were first isolated, or reports, written records are from Goa in 1979. So the research on mangrove yeast set in much later. And this is this very reason that there are not much studies, and the studies are rare, that we have taken up. And this is our paper, that is the diversity, distribution, and the bioprospecting potentials of Manglicolis yeast. You can please read up this article in Sense. And now, if there are students who are interested in studying yeast, then they would ask me, what are the samples from which I can isolate yeast? So let's see where and from how you would go about. Actually, most of the studies have been concentrated on the water and sediment samples from mangroves. But equally interesting and intriguing are the association of yeast with vegetation. Actually, every part of the mangrove tree, be it the leaves, the stems, the bark, the root, you find associated yeast, and also the large number of invertebrates that reside in mangroves. Some of the reports are from from crabs and conches and even shrimps and lobsters. Okay, so different invertebrates also, they have been isolated. So the usual sampling would use sterile sampling bottles or skin bottles, and you need a hand corer to collect the samples. For vegetation, you will require sterile blades or knives to cut samples, and then Ziploc bags to carry it to the laboratory. So I'm basically concentrating on, or my team is basically concentrating on yeasts from mangrove vegetation. So around 18 species of mangroves are all reported from all over India and in Kerala, especially in the central part, that is the mangroves I've been working on. We have been able to identify 13 species of mangroves, and we have isolated yeast from all these 13 species that have been shown here. These just to show the distinct appearances of these mangrove vegetation. When we look at the isolation techniques, both culture dependent and independent approaches have been taken up, mostly culture dependent. And the main constraint that we face in the culture dependent approach is that yeasts are basically slow growing. As they are slow growing, we often face contamination from bacteria. And even if there are larger fungi growing in, picking up yeast from a clay that is contaminated with a higher fungi is difficult. And most often rare species may not be depicted in culture. The media that is very commonly used is wicker hams and malt extract for the initial isolation. Often stocking is done in malt extract, and these media are incorporated with antibiotics like chloramphenicol to actually reduce the contamination from bacteria. But it is difficult. We cannot avoid larger fungi. So that is a major challenge that we face in isolating yeast. Then, based on how do you go for identifying them, you can use microscopic, biochemical, and molecular characterization. Barnett's reference is the standard. So you can see some images on your screen now. That is the microscopic images. On the left, you can see the wet mount of yeast, and in the center, you can see the simple stained yeast cells. So you can see how beautiful they appear. It's almost like a honeycomb appearance they show, and the difference in morphology. They can be elliptical to ovoid and different shapes. And on the right side of the screen, you can see the pseudohyphae that some of the strains form. And this is of diagnostic or identification importance, because not all strains or all yeast species show pseudohyphae. So that is the microscopic appearance. And coming to the cultural characteristics, often probably most of you would have seen the typical yeast, which appears creamish or creamish white in color. And another thing that is interesting that we observe, that during the primary isolation from the samples, that when the yeast start to form colonies in plates, it gives out the very beautiful fruity yeasty smell. So you know you have isolated yeast. So that is the intensity of the yeast smell that you get on primary isolation. And there are a large number of pigmented forms. The red yeast, which are carotenoid producing, and you also have black yeast, which have a melanin pigment. So that is how the cultural characteristics. And then biochemical characterization, there is a huge array of tests. And of course, purely based on biochemical, from our experience, it's not always possible to confirm. But yes, you can group them, you can cluster them based on their biochemical characteristics, and like using primer software, you can cluster them. And from each cluster, you may identify the representative isolates. And another major test that is to, because yeast basically belong to ascomycetes or basidiomycetes, the one way of grouping them to these two broad groups is based on the DBB, or the diazonium blue bead test, where basidiomycetes are positive for the test. They give you a red to reddish brown appearance when the reagent is added. And the others, the ascomycetes, do not give that typical. You can see some ascomycetes here which do not stain brown or reddish brown or even pink in color. Another way to distinguish them, or one of the main characteristic feature to group them, whether they are oxidative or fermentative. For that, you can use marine oxidative fermentative media or other oxidative fermentative media to distinguish whether they are oxidative or fermentative. And there are, like I said, a lot of array of biochemical reactions, like a lot of hydrolytic enzyme production, urease, nitrate, all that, which is very similar to that you perform for bacteria, can be carried out for biochemically identifying them. And the identification, that the molecular base of identification is basically based on sequencing the ITS region, and the often used primers are ITS 1 and 4. I've listed others here. And another new approach, of course, like in all other environmental samples, the metagenomic approach to understanding yeast diversity, mangrove yeast diversity. But however, studies in this area is very, very scarce. And what we personally experienced in going for metagenomic studies or metabot coding studies is that we found the isolation of the DNA from the vegetation very difficult because of the high amount of polysaccharides and polyphenols that interfere with the DNA extraction. I think that this area calls for a lot of more research and understanding how we can successfully, very easily isolate the DNA from the mangrove vegetation. Now, I would like to move on to the current studies that are happening around the world. This is as of January 2023. You can see, though I showed you an earlier map where the mangrove coverage was shown, you can see the continents of Africa and Australia, especially Australia, which has quite a dense and a large coverage of mangroves. There are no reports of mangrove yeast from these locations. Most of the studies are concentrated in the Southeast Asian countries like Taiwan, Thailand. Of course, in India also there are reports, and some countries like Florida, Bahamas, Puerto Rico, and Brazil from America. So this gives you a lot of gap areas in yeast research, and probably areas where youngsters or people interested in studying yeast can take up. Because there are a lot of areas that have not been investigated so far. Now, moving on to the diversity of yeast from these different continents, from the pie diagram, it is clear that the diversity of yeast, actually the diversity of mangrove yeast were believed not to be very diverse, but from our review and our base of studies, we find out that is not so, and there is more investigations to be done in this area. So you can see the case of Asia, where it is the yeast diversity is really high, whereas compared to the other continents. Well, this could be a reflection of the lesser number of studies from these regions. So this calls for further studies in these continents. And one thing we can very clearly see that Candida, Rhodotorula, and Cryptococcus are cosmopolitan in distribution and definitely a member of the autochthonous groups of yeast in these ecosystems. And some new genera that have been identified, mangrove yeast, are Geminia, Kazakhstania, and Vishnikosima are some of the new or latest new genera that have been of yeast that have been identified from mangroves. Now, I have told you the three major samples from which you can collect yeast, that is the sediment, water, mangrove vegetation, and invertebrates. So the Venn diagram very clearly indicates some of the, you can see those in the center are clearly a genera which are cosmopolitan in distribution, and some are host specific, as in the case of invertebrates, where Exophilia and Funaceae have been only reported from invertebrates and no other component of mangroves. In total, we've been from the review, we would clearly get around 62 genera of mangrove yeast are reported as so far. Now, moving on to the studies from India, those Sundarbans is one of the best mangroves in the world with the largest area of coverage. The studies on mangroves from this location is scarce. You have quite some studies from Pichavaram and Muthupettai in Tamil Nadu. Similarly, from Goa and Kerala, a few reports from Surat in Gujarat are also available. One report from Gujarat. There is Prasanna Kumar has made a extensive study to create a DNA barcode library of marine yeasts that have been derived from the coastal sediments in mangroves and so that is one barcode library that we came across, but we didn't see anything others. Reports we did not come across. And this is the area of my current study, that is the mangroves of Puduvai and Kumbalam. We have been so far able to isolate 25 genera and 45 species of mangroves from these two mangroves, with Puduvai being generally being more harboring more diverse groups of yeast. And when we look at from the different parts of the mangrove plant, we find the leaves harboring the highest density of yeast, or the yeast populations were highest in the leaves, and also the diversity also was greatest in the leaves, and the least from the flowers of these mangroves. Here too, Candida, Clivoromycetes and Rhodotorula are dominant genera. Actually, Candida tropicalis is a highly dominant or a predominant genera that has been isolated from mangroves world over. And we also find something interesting, that pigmented yeast being more in numbers in the phylosphere region of mangroves to other parts. So they could have some association with the upper parts of the plants. We also have had an opportunity to study the diversity of yeast during the 2018 floods, and we found Candida tropicalis being predominant, and it is found to be highly resilient to the floods, because they were isolated during the different, we had done sampling before floods, just after floods, and post-flood samplings were also done. And we found Candida tropicalis to remain dominant throughout the different sampling. And we also found that Hortia wernicke was isolated only during the post-flood samples. And this genera, Hortia wernicke, is actually black yeast, and they are actually found in hypersaline environments. So some genera like Rhodosporidiobulus was isolated only just after floods. So probably they were introduced as a part of flood water. So there are no comparisons of previous studies for us to authenticate, or studies after that. So these are some of the observations that we made from our flood studies. And moving on to the bioprospecting potentials, they have been listed in this figure. The immense potential for mangrove yeast. One of the major areas they are known for us, their capability to produce enzymes. So basically, being saprophytes, they are known to produce an array of hydrolytic enzymes that that are valuable tools which can be used for biofuel production, the textile industry, food and feed industry, and so on. So I've listed some of the enzymes that have been reported from mangrove yeast. Now, another area is biofuel production. Biofuel is the need of the hour, and as an alternative to fossil fuels, or Basidium melanogenum and Saccharomyces cerevisiae have been reported to have the capability to produce biofuels as bioremediators. Candida is topmost genera which have bioremediator application. Then some strains of Geotrichum have been reported to accumulate heavy metals. Then they are also known to produce a large number of antimicrobials, which act not only against multi-drug resistant bacteria, but also against fungi and even pathogenic yeast. They also have been reported to produce biomolecules which have anti-cancer action. Some other interesting biomolecules include xylitol, polybeta-L malic acid. Mangroves were found to be capable of green synthesis of nanoparticles. They are found useful as probiotics in aquaculture. And some of the areas that I am moving on to the areas that I'm and my team are working on. One is the pigments of these yeasts. We have extracted the pigment and we found to have strong antioxidant, antibacterial activity. Another area that we are investigating is the use of these yeasts as color enhancing feed additives or pigment enhancing feed additives in aquaculture. And we have used and prepared these feeds and made studies on koi carp and Dulcinea filamentosa, and we have got very, very promising results in this sector. And another area that I'm currently concentrating on is the plant growth promotion by mangrove yeast. Again, we have been very successful, and these are a list of some of our publications in this area. And another area that is on biosurfactants. We have isolated and extracted biosurfactants from 10 yeast. They were shown to possess antioxidant, antibacterial, anti-cancer, and hydrocarbon degrading potentials. But sadly, when we talk about mangroves, which are so high diverse and have hotspots of both macro and microflora, this is the current status of mangroves, I think, in most parts of the world and India as well. Deforestation, the accumulation of waste, in the name of developmental activities, these mangroves have been destroyed. And with these loss of these mangroves, will we lost the vast potentials, organisms that are present in these mangroves. So we on our part should actually take actions to do. And we never should end a story on a sad note. And I'm happy, and probably you would have all come across that the central government or the union government has recognized the importance of mangroves, and they have started a scheme called MISHTI, Mangrove Initiative for Shoreline Habitats and Tangible Incomes, which aims at promoting and encouraging the re-establishment and restoring mangroves along the coastlines of India, keeping in mind the socio-economic aspects of the stakeholders as well. It's a big step in the part of the union government, and I'm hoping that it will help in restoring these very useful ecosystems. So to conclude, I hope you will all agree from talks right from the morning, mangroves are a treasure trove of both macro and microflora, flora and fauna. And MycoAsias are largely unexplored resource of immense biotechnological potential and have the potential to play a key role in environmental remediation and sustainable development. I would like to acknowledge MycoAsia, MycoIndia for giving me this opportunity, especially Dr. Belay Damodar, sir, for having given me a chance, and the Kerala State Council for Science, Technology and Environment for the funds that initiated me into the studies on mangroves. This is my team. I'm standing along with my guide, Dr. Sarama, and the rest are my students. So this is the team that has been working in this area. I'd also like to thank Dr. Kadireshan, sir, who's been inspirational and a guiding force to us. And please free to contact me, some of our publications. And I thank you for your patient listening, and I'm open to questions now. Thank you.
Thank you, Dr. Manjusha, for the wonderful presentation on the marine mangrove eel. Can you hear me?
Yes, sir. Yes, sir.
Okay. I think I will invite the audience to ask questions. Ladies and gentlemen, if you have any questions, please raise your hand, and I will let you ask the question. This Danya Keshavan, please. Danya, okay, I need to unmute people. Okay, Danya Keshavan, please.
Ma'am, good evening.
Good evening.
Ma'am, I isolated my cultures from seagrass. I got some yeast also along with that endophytic yeast. In that, pink and black was a little bit prominent, I think. Pink and black.
Pink.
But
Okay.
during pink and black yeast. But while subculturing it, I am seeing that color, no ma'am, that pink color is getting lighten and lighten. And could you give any tip to retain the color while subculturing?
Okay. This issue we did not face despite isolating a large number of red yeast. Did you check the salinity of the medium in which you prepared the media? And in
Medium,
yeah.
sorry ma'am.
Uh,
Salinity is 15 ppt.
and
I'm doing all culture.
are you maintaining it in 15 ppt itself?
Yeah, ma'am.
Okay. So one thing, these pigments are actually carotenoid pigments are sensitive to light. Have you noticed any color change when you store them in the refrigerator?
Ma'am, I used to keep a working culture in the refrigerator. After that, only when I used to subculture, this color is getting lightened.
Actually, we observe the reverse. When we kept, for example, in the case of Orobasidium pullulans, when we first initially isolated, they give you a Coca-Cola kind of color. And the color deepens to that real deep black on storage. And we did not face the issue of a carotenoid yeast color fading. So that issue we did not face. So maybe you can try wrapping it up and covering up with, say, maybe a black paper or a black cover, and check out whether they are still losing their color.
Ma'am, one more thing I want to.
Have you confirmed their yeast?
Yeah, ma'am, there is.
Okay. So you may try that out, but if it is a pigment that is fading, I don't know whether you can use it for a pigment-based application if the color fades, because our pigment stayed on for quite a long time. I mean, the colors did not fade actually.
Ma'am, also one thing I want to ask is that black color yeast, ma'am, got
Yes.
no. I think I got a similar one, but it is not growing. I mean, is it a slow growing yeast?
Yeah, yes. Black yeast, especially if it is Hortia wernicke, this issue is there. We have not, even the first isolates, we were not able to, once you store it, it's difficult to subculture them. That issue is there with black yeast. Maintaining them is difficult. We lost around 3-4 cultures because of this issue. It is difficult to maintain them in our laboratory. That issue we faced. I think that has been reported also, that it is difficult to maintain Hortia, especially in culture.
Ma'am, one more thing I want to ask is that I have checked in GYP, nutrient agar, malt, and potato dextrose.
Okay.
Could you suggest anything, a better medium for culturing?
Is it for initial isolation or maintenance?
Maintenance.
Maintenance,
And
initial isolation, why don't you try Wickerham's agar? Have you tried Wickerham's agar?
No, ma'am. No, ma'am.
We found best results with Wickerham. The composition is almost similar. It is malt extract and yeast and all those things. But you try Wickerham's for initial isolation, and try maintaining, if especially with issues with pigmented yeast, try maintaining them in Wickerham's itself.
Okay, ma'am.
Or try malt extract, because we store cultures in malt extract.
Okay.
And where are you from, Danya?
Ma'am, Kiset.
Okay.
Dr.
Thank you,
Samir,
ma'am.
Dr. Samir Kamre, please.
Yeah, Dr. Manjusha, nice
Yes.
talk and nice work.
Thank you, sir.
Yeah, continuation with what you said about Hortia. I was interested, like you said, difficult too. So how are you preserving your cultures as of now?
So we have working stocks in the refrigerator. We have glycerol preserved stocks in the deep freezer, and we also have paraffin stocks. So these are the ways we are maintaining them.
Yeah, my
Liquid
suggestion
paraffin.
is, Manjusha, you should go for lyophilizing, because that way you'll be able to preserve your cultures better.
Okay.
Okay, and the second question I have with regards to your work is that, did you look anytime with regards to the salt concentrations for these cultures, what you're working with? Like, what is the highest salt they could tolerate while growing?
Sir, we have checked about up till 25% salinity. Most of the culture prefer around 15%. Most.
Okay, so you're not gone beyond 25 as such.
No, because we found the growth to drop beyond 25. So we checked up till 25%.
Okay, and have you done any molecular studies with regards to salinity tolerance?
In the aspects gene level study on
Yeah,
salinity.
Yeah,
No,
yes.
sir, we haven't gone to that extent. We have just done culture dependent studies, but yes, strains we have identified on molecular basis. We have around 215 isolates. All we have identified on molecular basis, the strains that we have. The problem is we don't have references, not much work on this area. So
No, there is one beautiful paper, long time back it has come. It's called as HAL family of genes. That's for salt tolerance. And
okay.
around 7 genes have been identified already. You should look into that for your
Okay.
high salt concentration growth cultures. It would be very
Sure.
interesting, because from India, nobody has reported anything so far.
Oh, okay, so that would be interesting. We will check up, because it kind of a highly intensive work. This would be specifically tracked towards salinity tolerance. Yes, thank you so much, sir, for that piece of information. We'll definitely see what we can work on in that area, sir.
Yeah, all the best, Manjusha.
Thank you so much, sir.
Dr. Samir, were you suggesting to increase the PBT more than 25% by any chance?
Yes, because I have few cultures which have grown up to 35 to 40. That's the reason I was saying. But those are isolated from salt pans.
Thank you for revealing it. Thank you.
Okay.
Yes.
Dr. Devadatta. Sorry to keep you waiting, Dr. Devadatta.
Very nice talk, ma'am. Actually, I have a question like, what are the best isolation methods for yeast? Because I found that very less yeast were identified or isolated. Very less group, very less people are working in yeast in India, especially from mangroves.
Yes, sir, that is the issue we are facing, because we have to compare or find out. One issue I will tell you is that, sir, we were practically, actually, basically, when we began, we were mostly interested in endophytic yeast. But the issue there again, because yeast, because they are eukaryotes, they are fungi. You cannot eliminate the contamination of higher fungi, though you can avoid bacteria by adding antibiotics. You cannot avoid higher fungi. And they're dominant. Endophytic fungi are dominant in vegetation. So if you surface sterilize the mangrove vegetation and try isolating yeast, then fungi overgrow yeast. But if you don't go for surface sterilization and you actually macerate the sample directly, just washing and in sterile water and then macerating, no alcohol treatment, and try doing it, you are able to isolate yeast better. And of course, the initial isolation takes time. You need to be, you have to incubate it for maybe 7 days or even more to initially get the isolates. But the indication, as I mentioned to you during my talk, when yeast start growing, it gives you that very strong yeasty smell on primary isolation. So when you get the smell, you know you have isolated. Then you have to work out, and like most people said, you need to pick it out, and as soon as possible the colonies appear, you need to pick it out and subculture it to avoid contamination from higher fungi that grow in these plates. Yes, I don't know. That is when this group discussion came on. When I joined this group, I asked Belly, sir, why is that nobody's speaking about yeast? There are talks on mushrooms and higher fungi, but nobody talking or discussing about yeast. So when I had asked him, and sir, actually, I think probably based on that, he asked whether I would give me a talk. So I don't know why people are not working. Probably they don't know. They're such fascinating group. I think more people should take up yeast.
Ma'am, have you tried the leaf litter of mangroves? So you found any good diversity?
I didn't get you, sir.
Leaf litter of mangroves.
Yes, sir. Actually, we have covered all parts. Leaf lifter also we have done, sediment and water. But today's basically I concentrated on the mangrove vegetation, because from mangrove vegetation, the studies are rarer.
Okay. Which substrate you found that you are finding most yeast diversity?
The leaves from the vegetation, it is the leaves, sir.
Okay. Thank you very much.
Thanks.
Madam Vaibhavi, please ask your question.
Yeah. Good evening, ma'am. It was a very informative talk. So, ma'am, I have one question regarding the salinity part. So, while the initial isolation of the endophytes from the mangroves, so is it like how much is it important to maintain the higher salt concentration of the media?
Actually, what we have done is, instead of distilled water, we have always used mangrove water in the media to avoid any other issues related to salinity. So from every sampling site, we used to collect the mangrove water, filter it, and sterilized it for preparing the media. So in that way, the salinity, the initial isolation issues can be avoided. Otherwise, we generally maintain it at 15 ppt, because the variations of ppt in these mangroves don't go over 20 ppt here. So that's
And
the
ma'am, in case if they are used, the media is prepared in distilled water,
Yeah.
how one can be sure, or how authentic could the endophytes be?
yeah.
Can they be the endophytic fungi?
Actually, that's what I said. My yeast, I don't claim them to be endophytes. They can be epiphytes, they can be ectophytes, because I macerated the entire leaf, and I told you I didn't surface sterilize the leaf. It was just washed in distilled water to remove the dust, and then it was cultured. Because we find when we look for endophytes after surface sterilization, the higher fungi overgrow, we are not able to isolate yeast. That is one issue we face. And of course, you can correct the PPT, you can prepare, like you prepare artificial seawater, you may prepare, or add maintain the salinity of your media to the PPT from where you're collecting the sample. That can be done if the water from that site is not possible. It will be ideal if you use the same water to avoid certain issues.
Okay, ma'am. Ma'am, actually, I'm working on this particular part where I'm isolating the fungi from endophytic fungi from the mangroves. So, as you said, I have used the artificial seawater agar as well as the water from the site which we had collected. So, I had these two medias, and also 9 different types of medias I had used for the isolation. So, why I asked you this question was, I had got culture isolates from the media using artificial and natural seawater agar, as well as from the other cultures, that is YME, yeast extract, malt extract, CDA, PDA, normal water agar, etc. So while getting these all isolates, and these isolates from the natural water and the artificial water, so are there any, I mean, can there be any problems within identification of those, that are they really endophytic fungi or no, basis on the basis of salinity I'm talking, because while doing, yeah, because
there shouldn't be, I believe there shouldn't be such issues, because you don't say they're endophytes basically because of their salinity tolerance.
okay, yeah, in regard of the mangroves, because we are isolating it from mangroves,
yeah,
that was
yeah,
my
yeah. Just because they the salinity factor, it is not it does not mean that you will not isolate an endophyte. It will be on your, like, you need to standardize your methodology. You can try out the media, check out in which media you're getting the highest number of isolates. Like sir said in the previous talk, how to check out whether your surface sterilization methods are 100% correct. Once you find that out, then you can even shortlist the media that is ideal for your purpose.
Yes, ma'am. What I had done during the isolation was the last wash, what I had done using the distilled water. It was the twice distilled water wash after entire sterilization. So I had, spreaded that last wash, 1 ml out of that wash in a PDA plate. So that didn't show any growth even after 20 to 30 days. So
So that should be fine, I guess. That should be fine.
so that showed that the
Yeah, definitely.
procedure, yeah, it was good.
Yeah.
So yeah. Thank you so much, ma'am. Thank you so much.
Thank you.
Thank you, Bobby. Last question from Dr. Mohan Kumar.
Madam, it was a really wonderful presentation.
Thank
My
you, sir.
question is, sir, what is the composition of the media what you used for isolation, madam? Because what is the percentage of carbon source there? So generally, the carbon source what is required for this yeast isolation, anything more than supposed to be 2 to 3%, and complex carbohydrate generally it is required. And what actually you have used in the isolation of the yeast, ma'am?
So the media, which is Wickerham's, though it's called Wickerham's, it is malt. It contains, like I mentioned, it contains malt, yeast, peptone, and the usual other ingredients which are present in other media as well. But it was actually, I believe, designed by Wickerham's for the isolation of yeast. So it's known another name as Wickerham's. So the ingredient, you mean the specific composition, if that you want, sir, the weight.
Yeah, concentration. But most of the times, this
Yeah.
this yeast and all that require cellulose, whatever content and complex carbohydrate. But whatever.
That's exactly I mentioned, sir. As I do not claim these yeasts are endophytes. They are associated with mangrove vegetation. They may be ectophytes as well. If you can refer to this paper that I have mentioned, all the compositions of all the medias that have been used for the isolation have been mentioned. So according to your work, you may standardize. We found Wickerham suitable. You may try out other media as well. The entire composition is given in this review paper in FEMS. You have all the medias that have been used. Only thing we need to incorporate an antibiotic to avoid bacterial growth, because otherwise bacteria will grow faster than yeast, so it makes your isolation a little more difficult.
Dr. Mohan Kumar, we are having sound issue. Please mute yourself. Thank you. Dr. Rohit Sharma, please, final comments for me.
Yeah. Thank you, Dr. Manjusha, for the very nice talk. I think you discussed very nicely about the diversity of yeast that Asia is having, and as well as the applied aspect of the mangrove yeast in the as a biosurfactant or a nanobiotechnology or agriculture or medical field. And thank you for pointing out the conservational aspects of the biodiversity. Thank you so much. So we thank you for taking your time out for the talk. Thank you so much.
Thank you, sir. Thank you, everyone.
Thank you, madam. Thank you, Dr. Manjusha.
So, moving
yeah, Rohit, let me continue. Yeah. Ladies and gentlemen, I hope I'm audible. Our next speaker is from Mexico. Dr. Patricia Velez. Can you hear me?
Loud and clear.
Yeah, thank you. Madam, thank you very much for accepting our invitation to talk today. Welcome to the International Online Seminar on Marine Fungi. And we are very much excited to listen to you and your work on deep sea fungi. Thank you very much, especially for taking the time on a Sunday early morning, 7:30, 6. I think I saw you at 6 a.m. Mexico time here joining us. Thank you very much for that. Before I hand over this platform to you, I request Dr. Rohit Sharma, managing editor of MycoIndia Academic, to introduce you to today's audience. Dr. Rohit Sharma.